Hello students, today we discuss the next question, that is the explain the role of various enzymes involved in prokaryotic DNA replications. So in previous video I already discussed about these prokaryotic DNA replications. In that there are about 10 different enzymes are used in this process, so they are having the different functions in different stages of these replications. So this question mostly asks for the short notes. So the first enzyme which is used in prokaryotic DNA replication is the DNA A protein. So the DNA A protein, it recognized to bind the ORIC region. So this ORIC region is the starting point of the replication. So this DNA A protein that are bind to this ORIC region, and then this the replication process is start. Then the next enzyme which is used in this replication process, that is the DNA-B, which is also called as the enzyme helicase. So this enzyme helicase breaks the hydrogen bonds. This hydrogen bond means these two DNA strands are joined by these hydrogen bonds. So these helicase enzymes do function as a breaking these hydrogen bonds. That's why the two bands or two strands of this DNA will be separated. Then the third enzyme which is used in this replication, that is the DNA-C. So the DNA-C will help in loading DNA-B. This also may to helps in the breaking the bondings of these hydrogen bonds. Then fourth enzyme is the Ssd protein. So these Ssd proteins will prevent the renaturations of DNA strand. So after the separation of the strands, sometimes if the SSB are not working, this DNA will be rejoined. So for avoiding this case, the situation, these SSB proteins are help to prevent this renaturations or recoiling of the DNA strands. Then fifth enzyme which is used in this replication process, that is the topoisomerase first or gyrase. This topoisomerase first enzyme will helps to remove the topological trace. Due to this formation of replication bubbles, the force are force towards the other DNA strands. So preventing this trace or force on the other remaining part of the DNA, these topoisomerase first enzymes are bind to this DNA on both sides, so the trace will be reduced of the DNA. Then next enzyme which is used in this process, that is the DNA-G, which is also called as the primase enzyme. These primase enzymes are synthesized the primers. So these these enzymes is used in the second process, that is the elongation. So in the elongations, for these synthesizing the primers, then only the new DNA strands will be start. So at first there is the need of this synthesis of primers. So these DNA G enzymes or primase enzymes are helps for synthesizing the primers. Then next enzyme which is used in this process, that is DNA polymerase first. So this DNA polymerase, these synthesized DNA strands in 5' to 3' direction, and that is leading and lagging strands formation. So after formation of primers, these DNA polymerase first enzymes are working and they form DNA strands in 5' to 3' directions, which is also known as the leading strands and lagging strands. That means some are continuous discontinuous strands and some are continuous strands are formed due to the help of this DNA polymerase first. Then next is the DNA polymerase third. This DNA polymerase third, and some corrections are in your notes. So this DNA polymerase third will help to remove the primers, which is also known as this exonuclease activity. So after the formation of the strands, the DNA polymerase third plays a role to remove the primers. Then DNA ligase. These ligase enzymes will seals the necks and formation of the phosphodigester bonds at both the ends, at the 3' and 5' end. So the function of the DNA ligase is to sealing the necks of the new form, जो DNA के जो न्यू स्टैंड्स फॉर्म होते हैं उसे नेक के साथ ज्वाइन करने का काम ये DNA लाइगेज करते हैं. Then last enzyme in this process, that is the topoisomerase 4. So this topoisomerase first separates the new DNA strands. So after the formation of new DNA, these two DNA are jointed. So for separation of these two DNA strands are done by the help of the enzyme topoisomerase 4. So these are the 10 enzymes which are working in the DNA prokaryotic DNA replications. So this is about the enzymes which used in the DNA replications. I hope all of you are got this. So you have to also mention with the help of this diagram. So in this diagram different names of the enzymes are placed, and in that actually which space they are working you have to mention in that figures. So in these first is the topoisomerase, then DNA polymerase third, primase, then DNA ligases, helicases. These are the different enzymes. You have to also mention with the help of the diagram. So I hope all of you are got these questions. So very easy, and they are most of time asked in the exam for the short notes. So now I stop here.